My name is Nianwei Lin. I'm the co-founder and president of at iXcells Biotechnologies. We are a San Diego-based CRO company and reagent company. As Amber mentioned, that some of you already become the old friend. I was there at Nashville with many of you guys. So today, my topic is disease-relevant cell models for rare neurogenetic disorders. This work was under a collaboration with many of our partners, including Mazi Therapeutics, Rare-Base, La Jolla Lab, and this work is not it will not be possible without the sponsor by your foundation. So I will firstly briefly introduce my company, iXcells, and with our expertise in disease modeling. And the second part of my talk will cover some of the preclinical studies around the N-of-1 ultra-rare neural disorders. And finally, I'm going to spend most of my time talking about the collaboration with Mazi Therapeutics and SLC681 Connect. About with the focus on the cell models. Our company is based in San Diego, as I mentioned. We founded in about 9 years ago in 2014. We specialize in providing preclinical solutions to pharmaceutical companies with focus on disease-relevant cell models. Particularly, we are a CRO and reagent company providing the off-the-shelf cell biology reagents, including the induced pluripotency stem cells, or iPS cells, and together with iPSC-derived cell models. With all these, we also have a large collection of primary cell isolated from the normal and diseased donors. With these disease-relevant cell models, we also provide our clients with preclinical CRO services, including the cell engineering, cell-based assays, as well as drug development, using the patient-specific cell models for personalized medicine. This slide is a good summary of our disease model. We offer two different major cell models, iPSC and primary cells. So besides these ready-to-use cryopreserved cell products, we also help our client to make their own disease-on-the-dish models to accelerate their drug development. So we help them with the iPSC reprogramming, gene editing, and targeted differentiation, including human neurons. These cells can be used for applications such as toxicology studies, drug screening, phenotypic screening, particularly, and also a variety of the functional assays. We have a very strong scientific team to make a reprogramming from patients' fibroblasts or blood samples. And after that, we have a full comprehensive QC criteria to characterize those cells. And iPSC-derived cells is another big category of our product line, with the focus on the neurodegenerative disease and neurogenetic disorders. We made a lot of different neuronal cell types, including cortical neurons, GABAergic neurons, which will be used in our SLC681 studies, and also sensory neurons, skeletal muscles, microglias. So with the background, I like to talk about our work with some of the rare disease foundations in the past 2 years. So with our expertise in cell modeling and RNA therapeutics, so we've been working with many, more than 20 different foundations to develop the individualized drug development. The modalities including antisense oligos, AAV-based therapeutics, trying to deliver messenger RNA, CRISPR, or base editing. We're also using the FDA and some clinical approved small molecule libraries for drug repurposing. And in terms of the cell lines, we're using majority is using the immortalized cell lines and iPSC-derived neurons, which I will be presenting later. And this is a timeline we proposed. And this, in the past 2 years, we've been trying to establish a standardized procedure for antisense oligo screening using the patient-specific cell models. So the procedure includes the cell model engineering, including the iPS cell line generation and differentiation into neuronal cells. We also did the gene expression analysis for antisense oligo screening, and followed by the in vitro toxicology studies. With our collection of a large collection of primate cell types, we'll be able to do the immunoresponse assays using the immune cells provided by the patient. Another areas we're trying very hard is trying to fill up the gap for in vitro efficacy or functional assays. I can talk about that later. So we try to use 3D and 2D models, trying to generalize, to make a general epilepsy cell models to see whether the drug has the real effect on the patient before we move on to the animal testing. And finally, in past 2 years, we have established a collection of validated reagents, including cell lines and control ASOs. So this is the timeline we propose to the community, although this timeline can vary quite a bit from project to project, from patient to patient. But overall, this estimate was based on the publication in 2018 for Milasen's. Many of you probably know Milasen's story, right? So from diagnosis to patient application, it only takes 10 months. So this is a very ambitious goal. But overall, so this paragraph show you a example how we help the patient to plan their strategies. So Excels can really help, starting from the cell line engineer. iPSC generation take 2 to 3 months. Neural differentiation take another 2 to 3 months. But before we have those patient-derived models, we can start to design antisense oligos and using the immortalized cell lines for the primary assay development and primary screening. After we have the hits, we can use patient-specific neurons for the hit confirmation and optimization and downstream functional validations. And this slide is this table summarizes the comparison of different cell systems we used in-house. We suggest our clients to start with the immortalized cell lines because it's relatively cheap. The cost will be a lot cheaper compared to the neurons with the large number of ASO. So after you get several hits, like 10 to 20 hits, you can switch to iPSC-derived neuron and for the hit confirmation and validation. Usually after identifying the hits, most of our clients move on to the animal tox studies at a GLP level. So those testing are super expensive, so putting a lot of financial burden on the foundations. And further, additionally, those gene expression, there's no clear correlation between the gene expression and the disease phenotype. So in order to fill in this gap, we are currently working with several research organizations trying to establish a in vitro efficacy or functional testing. So this slide is showing a co-culture system combining the iPSC cortical neurons. This is a glutamatergic neurons and astrocyte. We tested using a multi-electro-array assay or MEA assays to measure the electrophysiological activities in the neurons. So this is a recording of the MEA. So on the left-hand side is a neuron from a normal donor, from a healthy donor. On the right-hand side is the neuron from a patient carry a mutation in a KCNQ2 gene. KCNQ2 is a potassium channel subunit protein. So the mutation causing a loss of gain of function. So basically the mutant allele block the normal wild type allele. So the strategy is trying to block them to bring down the allele specific mutation by antisense oligos. So because we already have the hits, so now we're trying to see whether by adding those antisense oligo, we can not only reduce the mutation gene expression, but also we can change the phenotype. So this is some quantification data by MEA. So you can see very clearly that the red line is the mutation. The black line is the wild type, is the healthy donor. So you can see the pattern is very, very different. We call this the enhanced phasic bursting. So this is very similar to the epilepsy symptom. So hopefully we can recapitulate the patient's disease phenotype in a dish. So this table summarizes a list of disease and patient foundations who are currently working with, and some of them already completed. So you can see a lot of these patients carries a rare neurogenetic disease. So many of these are ion channel. Also, you can see SL66A1 here. This is just the antisense oligo project where we have different numbers of ASOs to start with. And in terms of the mechanism or MOA, we have allele. We try to use allele specific. Also, some of them are allele specific knockdown. Some of them just general gene knockdown. And we also have several projects trying to increase the gene expression, including SL66A1. So this is more challenging. And this is the list of the projects trying to use a different modality. So the collaboration with Rarebase is trying to use the FDA-approved library, small molecule library. And the collaboration for Rett syndrome, the project for Rett syndrome is trying to use base editing. These are still ongoing projects. And we also have a list of patient foundation. We're trying to help them to generate iPSCs. So I saw combined brains here, so we can talk about how to collaborate to make those cell lines easier to access, accessible for a research community. So we do have we often be asked whether we can become a repository to ship those cell lines to different research labs. But I think we should not recreating the wheel. By collaborating with different organizations, we'll be able to better distribute those cell lines research tools for the whole community. So we've been partnering with a lot of different biotech company, patient foundations, and you can see a lot of, probably some of you recognize some of these kits and foundations. So we also work with some nonprofit organizations and research labs, including Timothy Yu's lab from Boston Children's Hospitals, who developed the first individualized antisense drug, Milacin. We also work with N=1 Collaborative. So if you guys are aware of the Milacin's story, so Mila passed away 2 years ago, and her mother, Julia Vatero, started to start another foundation called N=1 Collaborative. It's a patient-driven nonprofit org. The mission is trying to standardize the individualized drug, particularly antisense oligo drug development, by putting all these building blocks together. So we are very, very honored to become one of the trusted partners as a CRO partners in this ecosystem. Their motto is from Mila to million. We are trying to standardize the procedure so that the patient came to us, they know exactly what to do instead of they spend tons of time to find out all these resources. All right, I think I'm on time, so I'm gonna spend most of my time talk about the SL661 available cell models. Again, this is a collaboration with Mazi, Rarebase, La Jolla Lab, and of course the sponsor, SL661 Connect as a sponsor. So Mazi Therapeutics have developed 3 strategies. Their therapeutic approach is mainly from 3 different angles. One is to target the long non-coding RNA or link RNA, which is one of them localized close to the SL661 gene. The second approach is microRNA masking, and the third one is the splicing regulator. So I'm gonna talk about the first 2 in my talk. This is a gene location of SL661 in chromosome 3. You can see the red arrow is the gene, and on top of it, you can see a small reversed gene called SL661 AS1. It's antisense 1. So that is a long non-coding RNA. So this is a gene doesn't have any protein as a product, but the research shows that this gene can function as the antisense to block its neighboring genes, which is SL661. So the hypothesis is trying to knock down this long non-coding RNA so that this targeted SL661 will increase. And this link RNA shows the highest expression in the brain tissue. But we realize that even they are highest, but still compared to other genes, the expression is very, very, very low. It's causing quite a lot of trouble to us. The second approach is the microRNA masking. Similar to link RNA, microRNA also demonstrate inhibitory effect on its neighboring genes, which is the SL6A1. This is a gene location. You can see there's a lot of microRNA locations. The strategy is trying to screen these microRNA to identify the one which is putting the inhibitory effect on the target gene. And we can design the anti-mir to bring down these blockers so that we can increase the target gene. So the first step is trying to identify good cell models. We all know that SL6A1 is expressed in GABAergic neurons. So the mission 1 is trying to screen a variety of different GABAergic neurons from different vendors. So we got so we're testing different sources of the cells from of course IX cells and BitBio, BrainX cells, Fujifilm CDI, and Lonza. Because this is a pilot experiment and the timeline is pretty tight. So Mazi decided to to get cells from different sources, but unfortunately, we don't have the same timeline for each cell. For example, we have the egg cells, brain egg cells, and big bio cells culture in our lab at day 7, day 14, and 21. But we can only have the frozen cell pellets from other sources provided by La Jolla Lab and New Site. And we're putting all these samples together, trying to see whether we have the gene expression in these cell lines. As I mentioned, we only have 2 cell lines that can be cultured in-house at the same time. So we compare the in-house GABAergic neurons developed by my company, and another one is BitBio's GABAergic neurons, which is generated by a different way. It's an inducible system. But you can see after 24 hours post-thaw, these neurons develop starting to build a neuronal network. And the viability lasts for extended culture time, which is more than 14 days. Actually, we culture for more than 30 days. The cells can still survive. And we further did a more categorization of these cells. So this is our in-house. We're going to launch this product next year, but when we start this assay, we don't even have those data ready. So we only did 7 days. So these are, I believe these are, it's hard to see from here, but we have 14 days cells. And you can see the synaptic marker that it's VGLU1 is the synaptic marker, and GAD67, that's a very specific GABAergic neurons. And MAP2 is a general mature neuronal markers. And so again, you see MAP2, TOJ1, those are very typical neuronal markers. And we are very excited to see PAVA booming, which is a PV on the left-hand side. That's a red signal. And we're very excited to see that because that's a very, very traditional inhibitory neuronal markers. We don't see it too often in the iPSC-derived neurons, and we see it in our cells. And to further validate the functionality of these GABAergic neurons, we test the GABAergic release by ELISA. So here, iPSC and DA neurons and glutamergic neurons is served as a negative control. And you can see our GABAergic neurons show the highest release of GABA compared to another source. And this is the MEA testing. I don't know how to play the video, but basically you can see this is the neuron, the pure neuron culture without an astrocyte co-culture. You can see the neural firing over the time. You see the increase, the bursting, and the number of the spikes. So as I mentioned, in this study, we used the day 7 neurons made by IX cells. So the gene expression may not be ideal because the cells are not mature yet. And finally, this is a gene expression in different source, the GABAergic from different companies. Overall, we are very excited to see the neuronal marker, GAD1, MAP2, all highly expressed in all these GABAergic neurons compared to the HeLa cells. HeLa cells is the green ones. And in terms of the SL6A1, we see the highest expression in day 21 neurons provided by Fujifilm CDI. But we see a decent expression across different vendors. So in summary, so we identify one of the good model for the downstream applications, and we can potentially. It's very clear that we see we see the maturation-dependent gene expression increase. For example, this is day 60 and day 90, same gene, same cell lines. You see the increase of gene expression. Just be noticed that this is the log 10. So actually, there are about 2 to 3-fold increase, just about 2 weeks apart. So we decide we will potentially use a more mature neuron in a future study. And because our cells, X cells, GABA-G neuron, we validated in the functionality assays by MEA and GABA release. So we think it's potentially is worth to try a different time points to see whether it can be also included in the downstream experiment. And another approach, another plan is trying to use the ovarian cancer cell lines, SKOV3. So this gene, we already know that SL661 gene is expressed in this cancer cell line, and cancer cell line is much easier to handle, so the cost can be much lower. So we decided to use these cell lines for microRNA screening. So again, there are 2 immediate actions. One is to target the long non-coding RNA. So we're gonna check whether this long non-coding RNA is expressed in some of the GABA-G neurons we selected. And once we confirm the expression, we're gonna design an antisense oligo to target this antisense, target those long non-coding RNA. For the MIR approach, similarly, we're gonna screen 13 different MIR. So basically, we ordered a MIR mimic, and we're trying to see whether this gene can knock down the SL661 expression in the cancer cell line. And then we design an antisense to target those MIR. All right. Finally, I'd like to thank all of our collaborators, Marzi, Rarebase, La Jolla Lab, and of course, Amber. You've been always the power of our studies. So we have about 12 PhD level scientists involved in this project. And I'd like to thank our project leaders, Jung Chao and Yu Chen. They are the main force of this project, and also the collaboration across a different team under our in my company. So with that, I'd like to thank you for your attention, and I'd love to have more discussion with the folks in the room later today. Thank you.